Well, thanks everyone for coming. Thanks to the organisers for organising. I'm sort of working across multiple projects at the moment in Oxford and in UC Davis and the Smithsonian. But I thought today I wanted to talk about, maybe kind of reflects what I do, which is a bit of a mixture between sort of method-y type stuff and empirical work. When I talk to biologists, I tend to be a computational biologist. When I talk to computer scientists, I'm definitely a biologist. So I want to talk about two questions. The first is really a fundamental issue about how do we define these associations, which I thought might be interesting to some of you to hear about or discuss. And then the second half of the talk, I'll talk about what are some of the implications of group structure. And Darren actually really nicely introduced a lot of stuff. Can't hear you. Okay. Try and speak louder. Darren nicely introduced quite a lot of stuff I'll talk about today, especially questions about assortment. So I want to talk about conceptual issues with network inference. I'll talk about scaling up methods, existing methods for dealing with bigger data. I'll talk about linking back what we find in terms of the networks back to informing ecological questions, and maybe a few reflections on where I think we might be going. But what I'm not going to talk about is any equations or math, and I'm really not going to detail any models. Sorry. So the first question, what is an association? How do we define our associations? Well, you know, when we're observing animals, I observe flocks of color-banded birds, like Dave does, and it's actually it's quite straightforward usually. Usually you walk through space and there's a group of birds, a flock, or whatever, and then around them there's nothing, and then there's another one over there. But as we get more increasing data and data becomes more complex, it can be harder to define. So if you look at the inter-individual distances, the distribution of these tend to be continuous rather than bimodal. So there's been quite a few common approaches. I'm not going to review them. Just these were taking them out of the top of my head. Some of them, such as spatial proximity, so like 1 over distance, which creates just a totally connected network. And then maybe you might threshold that distance at a certain distance. And then there's some group sort of measures, so more akin to when we do observations, which is sort of looking at time or spatial windowing. I'll go through these a little bit. So threshold approaches, so you could look at connections based on the distance between pairs of individuals, or as Darren said, you could take a certain number of body lengths away and so on. So that might be okay for some networks, and here I sort of, after chatting with Meg this morning, I thought I'd put a little disclaimer that in some cases it might be really biologically relevant to use these networks, these thresholds. So for example, if communication only goes for a certain distance and you know what the distance is. But it can be an issue for just defining association groups when the structure might be more fluid. So for example, when groups kind of mix and merge, when there's fission-fusion dynamics and so on. So for like example here, you might have a group of what is really red individuals and a group where what is really blue individuals, use this color codes throughout, and then at some point like they sort of come together and there's some individuals that are mixed across both groups and it's unclear where they are. But perhaps less of an issue, I think that's maybe less of an issue, might be more of an issue, is that the scale of the interaction can change. So it can be influenced by some ecological parameters, such as the density of the habitat. You might find that individuals are closer together when they're in more dense habitat, and then spread further apart. So your one threshold does not necessarily have the same biological meaning across all the contexts in which you apply it. And finally, as far as I know, I haven't really seen anyone really try and address this issue with, especially with sort of these tags that look at the distance and so on. So these really dyadic measures of association is whether associations are transitive. So for example, if A is connected to B and B is connected to C, do we then infer that A should be connected to C as well? So I thought I'd sort of revisit maybe whether the gambit of the group is actually a meaningful approach, maybe an approach that might become more useful again as we get more and more data. And also because I think that groups might have some important properties that we maybe don't capture with really dyadic measures. And, well, I can't remember what I was going to say about that point, so I'll just skip it. But really, I mean, one of the points is actually that one of the reasons why we kind of, I think, as network people, my impression was they sort of threw out the gambit of the group was because of the impact of sampling error and so on that you had on it. But as we get more and more data on every individual, I think that the issues of sampling errors or sampling biases can really be dealt with quite well. So then how do we identify groups? Well, typically you can use windowing approaches, which is you might say every individual that I've seen in this time period or in this space I allocate to this group, and then I'll take the next one, allocate to that group. But that can have some issues. For example, you might have two different groups, like there in T2, you might have two different ones that get lumped together. Or perhaps more importantly, it can lead to splitting of groups. So for example, if a group occurs in the boundary of two observation areas. But the way I kind of approach these things is always think about what's the definition of what we're trying to observe. And the definition of a group is a number of individuals that are located or gathered or moved together. So really, to me, the answer for that is to actually look for non-random clusters of individuals in space or time. So cluster analysis is a really common method in this common machine learning method in used for lots of different things. There's lots of different algorithms, most of which I don't understand. And but essentially, my idea of them is that they exploit the non-homogeneous density profile, non-homogeneous distribution of particles or individuals or whatever it is you're measuring. And I think distribution models are useful here because individual clusters, often when animals are found in space, they might be normally distributed, so there's like a center and individuals around or something like that. But most importantly is they don't assume a priori constant or constant interaction radius, so they can be really flexible. And some of you may have come across this work mostly in Jens Krause's review, but the original paper on this by Yannis, who's part of our group in Oxford, where we work on great tits, he wrote a paper about why we should use, or how we can use Gaussian mixture models to identify clusters of birds visiting feeders in time. So these birds are fitted with PID tags, and they visit RFID feeders that are in Whiteham Woods. I'll talk about that a little bit more, but here I'll just give you an example. So this is one of our feeding stations. There's two antennas, and the birds come and pick up the seed, then fly out. The cage is because they get absolutely trashed by squirrels, so we have to protect them from squirrels. You can see they sort of, the flock comes around and they each come and feed, but they're not all at the same place at the same time. So they create a sort of a temporal stream of visits. Yeah, go on. So this sort of data stream, as I said, really what we want to try and infer from this is that we have some event, or what he calls gathering events, that are in space, but they might be different sizes, different lengths. They might be very close together as well. And so what Gaussian mixture methods do, his package that's in MATLAB does is it actually identifies these clusters and creates a bipartite network of individuals to events, which is essentially a group by individual matrix, and then we can use that for doing all sorts of network stuff, which is great. And they can also, be assigned to time and place and so on. And so in Whiteham we create these wonderful networks of great tits. This was work that we recently published on personality and great tits. But this sort of structure really facilitates doing permutations and using null models using permutations, and you can do standard permutations of your network, and then you can also do the permutations of your data stream or group permutations. But before I talk about that some more, I also want to talk about the fact that I think this is really applicable to GPS data. And so, for example, this is just a demonstration of using clustering on just randomly spaced individuals that had a center point and then Gaussian distribution around those points. And so some of the work, come on, look. So this is what I'm working on largely at UC Davis with Meg Crowfoot, where we collared, or she collared, these all the adults in a troop of baboons, and with GPSs at 1 second resolution, and you get this sort of distribution of individuals moving around in time. This is at 15 times speed. And using Gaussian mixture models, I simply inferred the association groups, and you can find the clusters of individuals in space. And what we're interested in doing here, actually, with these data, is very much along the lines of what Noah was talking about, and other people. Is really understanding the link between the dynamic social structure and processes of within the troop, such as how do troops make decisions, how do they navigate through space, and how different individuals might play different roles in this, and questions about leadership. Anyways. So now that sort of brings me on to the second half of my talk, which is really thinking about what are the ecological implications of group structure, and how maybe do we infer what's going on from these data. So how can we use social networks to understand what the mechanisms that underpin population demographics are? So as Darren talked this morning when he was talking about the cooperation thing, how do you understand what are the mechanisms behind assortment in social networks? And so assortment is really the preferential attachment of similar nodes together. And it can have some, it can have some really important consequences for fitness through the interactions of phenotype. So for example, the easy one to think about is the confusion effect. So if every individual is of exactly the same phenotype, they might confuse an incoming predator, but if one of them is a totally different phenotype, then it's going to suffer a fitness cost by being easier to target. But also, some of the work I'm doing on this is looking at questions about social selection, so how your selection pressure is influenced not just by your phenotype's interaction with the environment, but also your phenotype's interaction with other individuals in your area that you interact with. And as well as cooperation, I think there's several mechanisms that can really drive associations. So one of them is spatial disaggregation. So for example, if small fish go into shallow water and big fish go into deep water, you're going to get assortment by body size. But also it could be by social decisions, so choosing who you associate with based on their phenotype within your area. And just to plug my recent paper, you can now measure assortativity using weighted networks. But how do we test for these competing mechanisms? And I think this is where one of the really nice things about networks is we have some really good null models. I think we can use these null models to get at some of these questions. So for example, we can ask whether assortment is driven by spatial distributions, sort of repeating myself here, or social affiliations, where different phenotypes are associating together in space. This is the outline of White and Woods. And I think there's sort of two null models we can use. One null model is we can do a node permutation, so we have our observed network, we think our network is a true representation of what's happening, and we can shuffle the identity of the nodes. So we allow individuals to be moved around in space. And if we reject this, then it suggests that what we observed in a population or in a subsample of the population are individuals that are non-randomly distributed in space with regards to the phenotype that we're interested in. A second null model is we could do group permutations. We can actually permute our data stream, and we can control for things such as space. So here we have an observed network, and then we just randomize individuals' observations, but we randomize it within where they were observed. And if we reject this, then it suggests that phenotypes are non-randomly associating in space and time. So now we have one model that we can, if we reject that, then it suggests spatial, and then the other one, if we reject it, it suggests more local effects. So in White and Woods, we collect a social spatial structure of, we look at the social spatial structure of great tits. What we do is we in the winter, we have 65 of these sampling sites which are randomly spread throughout the woods there, those white dots, and they all open and shut at the same time every week. So you basically get a snapshot of the birds that are fitted with pit tags. There's about 1,000 grey tits that live there, and these exhibit really strong fission fusion dynamics. This is just a really simple measure of stability, which is based on how many individuals when you're next observe, how many individuals are the same in your group, and this is just a ratio with random, and you can see that in the first 10 minutes it's really highly non-random, and after that it's only just above random, and that's probably the work we're doing now suggests it's actually driven by partner choice and pairs of partnered individuals. And what we wanted to find out is whether the social network is structured by questions such as age, or immigration status, or dispersal, and this is sorted by sex, so that whether we can really find these mated pairs in our data. So I'm just going to quickly give an example of how asking the question of how population is structured based on this immigration status. So this is whether birds. What we define as immigration status is birds that are residents are born in a nest box in White Woods, and birds that are immigrants are born outside of White Woods and come into the woods, which is about 50% of the population usually. And so when we had our observed data, so this is not a very clear graph, but the black line here is our observed data. This is the 95% confidence interval of the node permutations, and this is the group permutations. You can see we really clearly, for small groups here, we really clearly rejected the node permutation, but we had very much the same pattern as the group permutation. And on the y-axis is the proportion of residents. So it suggests that there's fewer residents in small groups than expected by chance. And when we look at the distribution of birds across the wood, we can look at the distribution of where all the immigrants are and what the population size is. You can see that actually the real hotspots for immigrants are areas where there's fewer birds. So what this is suggesting is that birds that are coming into White Woods are actually avoiding these really high population area, high competition. Anyways, areas where there's lots of competitors, and they're going to areas where there's less competition, and we can go back to some of the literature and find that this is probably due to resident effects. The resident birds tend to be more competitive. Even resident juveniles can be competitive over newly incoming adults. And doing this across different things, we found that in this case, the age was really driven by social effects, so really local effects. So the composition of groups was determined by the decisions that the individual made within the local area, and same with sex, whereas this immigration status was some kind of spatial. So it suggests that there's different scales of decision making operating here. Actually, I'll say spatial, but it's probably still a social decision that's made on a spatial scale rather than on a local scale. So why do I think this is important? Well, I think selection operates on genes. And as I said before, I'm interested in this question of social selection, such as interacting phenotypes and how that can impact the selection that is operating in a population. And also there's potential for social niche construction and so on. And most importantly, everything that I found here existed despite the population being really highly fluid and lots of fission fusion. So where do we go from here? Well, I think that the methods are somewhat keeping up with data collection. For example, we're coming up with methods for looking at dynamic networks, network inference, models, and so on. But I think our data collection is somewhat exceeding our ability to test hypotheses and formulate hypotheses. Particularly, I'm interested in questions linking structure to processes, which was really nice to see lots of people talking about that already, and questions moving beyond the dyad. So for example, questions of self-organization and collective behavior. How does population structure emerge from the individual decision making? So none of these things are individuals making decisions randomly. They're making decisions with respect to what other individuals are doing. And are there certain social structures that are adaptive, and do they lead to emergent group properties? Some of the stuff we're looking at with social learning and White where we are looking at the spread of innovation and so on, is targeting these sorts of questions. And actually, talking about this this question of comparing networks, we've got a paper as well in review at the moment, not comparing networks, but for those of you who are familiar with network-based diffusion analysis, I've just been working with Will Hoppett, and we've come up with a new way of testing whether diffusions can comparing networks through diffusion analysis. So you can provide multiple candidate networks, and you can look at whether they have the same rate of diffusion or whether they have different rates of diffusion. And just for a bit of fun, I thought this is what I mean by social. So you can find this on YouTube. Most of you have probably seen this, but these randomly occurring particles can come together, create structure, and then have some emergent property. And this keeps going for a while. And so we're working on a lot of these questions, especially with the baboons, with this really great data set. And there's a few of us here today, Tanya, Meg, and Ari, and we've got a poster downstairs, and if you want to talk about it, please come and see us. Finally, I just want to thank people in EGI in Oxford that I work with on the birds, the team of Bionic Baboon team, and the organisers here for inviting us. And that's it for me. Thanks.